So yes, very good afternoon to all the students of BPC. So here we are looking into the questions, short answer type of questions from histology and anatomy of flowering plants and modes of reproduction of your BPC first year students. So here if you look into obviously we are expecting most of the times from histology and anatomy of flowering plants, most of the times you will get LAQs, okay, but then there is equal possibility of short answer type of questions as well. So look into that with utmost care. First is about histology and anatomy of flowering plants. You will get at least 2 questions in your EAP set exam if you can see all the EAP set PYQs. And if you can consider NEET also, definitely there will be at least 2 to 3 questions from histology and anatomy of flowering plants, 2 questions at least, okay. So that important this chapter is. So whenever you are going through this chapter, you just don't mug up the concepts, okay. Don't just mug up the concepts for IPE examination, just have a sequential preparation, so that you will be ready with the complete content and you will be ready for both subjective and objective type of exams, okay. So here we go with the first question, state the location and function of different types of meristems. What is the location and function of different types of meristems? Just a second. I hope now the screen is clear to everyone. Yeah, now state the location and function of different types of meristems. This has appeared in multiple years, like if you can see in March 2024, 2023, 2019, 2017 and 2016, 2015. In every consecutive paper you can see this question. So here if you observe beta, first location and function of different types of meristems. First is apical meristem. Apical meristem location, if you see, these are present at the tips of roots and shoots. Apical meristems, meristems that are present at the tips of roots and shoots. Main functions, if you see, they will produce primary tissues, formation of primary plant body, and they will promote linear growth, which will help in the increase in the length of the plant body, and they will form axillary buds that will give rise to the branches and leaves. Okay, this is about the apical meristems. And then if you consider this diagram, very important diagram. First let me explain you these 2 diagrams about root apical meristem and shoot apical meristem, which will be clear for you both for MSET and NEET and IPE exams. So these 2 diagrams are very important for you people to present in the exam. If you are choosing this question for SAQ, it is mandatory to present this diagram in the exam. So here if you consider, first is root apical meristem followed by shoot apical meristem. So root apical meristem, if you observe, root cap is at the terminal end, as root cap is at the apical region, root apical meristem which is present over here above the root cap, root apical meristem is occupying the subapical or subterminal region in this, okay. Root apical meristem will occupy subapical or subterminal region. Subapical or subterminal region, and close to the root apical meristem, as root apical meristem is the hot spot or hub which is filled with the initial cells. Okay, close to the root apical meristem you can see initials of central cylinder and cortex, which will build up the central cylinder and cortex region, and later they get mature to both cortex regions, and at the center you can see central cylinder. At the center, central cylinder and cortex, these are developed from where? Initials of central cylinder and cortex. And at the above region here you can observe, okay, at the peripheral end, which is known as protoderm. Protoderm later matures to what? Epiderm. Okay, protoderm matures to epiderm. Fine, right? And here you can see initials of the capsules which will develop the root cap. Clear, right? You remember one thing with respect to this diagram, that root apical meristem is present at the subapical or subterminal region. Close to the root apical region you can see the initials of central cylinder and cortex, which will later mature to the central cylinder and cortex, and below this you can see initials of root cap which will give rise to the root cap. Clear with the root apical meristem, ma? Now we will look on to shoot apical meristem. Unlike root apical meristem, shoot apical meristem is present at the apical region, or it is present at the apex. This is present at the apex or apical region. Okay, above this, in close proximity to the shoot apical meristem, you can see leaf primordium. Leaf primordium is present in close proximity to shoot apical meristem, and below this you can see axillary bud. Axillary bud can give axillary bud which is formed at the axil of the leaf. Okay, axil is the angle which is made in between leaf and stem. Okay, at this axil region what is formed? Axillary bud is formed. Okay, axillary bud is formed at the axil region. Just a second. Yeah, at the axil region axillary bud is formed, and this will have initial cells where this can give rise to a floral bud or a leaflet, or it can give rise to a shoot bud. Okay, this can mature to any of this. Fine, right? And in close proximity to the axillary bud, you can see differentiating vascular tissue. Differentiating vascular tissue is present in close proximity to the axillary bud. So, this is the complete information about root apical meristem and shoot apical meristem. Now, we will move on to intercalary meristems. Intercalary meristems, where are they present? So, when you are describing each type of meristems, once recall the flow chart which I have given you in your EAP set and NEET classes. I have given you a clear flow chart, like how meristematic tissues are classified based on the position or location and based on their origin. Based on their origin, if you see, we have primary meristems and secondary or lateral meristems. Again, based on the positions, primary meristems are differentiated into apical meristems and intercalary meristems. Apical meristems are present at the apex regions of root and shoot. Intercalary meristems are present in between two nodes in the intermediate region. Clear, right? So, this is intercalary meristem. Okay. So, location of the intercalary meristem, if you see, which is present at the base of the leaves. This is present at the base of leaves at internodes between mature tissues. This is present in between the mature tissues. Functions, if you see, this helps in elongation of internodes. Okay, this helps in elongation of internodes and rapid linear growth. This helps in elongation of internodes and rapid linear growth. This aids in rearrangement of parts which are removed by grazing herbivores or damaged. Okay, which are removed by grazing herbivores or by damage. And these are common in intercalary meristems are common in grasses and monocots. If you see lateral meristems, the location of lateral meristems, if you observe, these are found in mature regions of root, shoots, and they are parallel to organ surfaces. For example, if you observe vascular cambium and cork cambium, these are found in mature regions of root, shoots, and they are parallel to organ surfaces, like for example vascular cambium and cork cambium. And the functions to be highlighted for lateral meristems include, what are the functions of lateral meristems? They will produce secondary tissues which will help in increase in growth of the stem, secondary growth, and this will help in the formation of wood, bark, and this will help in strengthening the tissues. Then you see, this will maintain the cylindrical growth of the organs. They will maintain proper cylindrical growth of the organs. And then you observe next second question, if you see, this is the complete description of both types of meristems, primary and secondary. Primary again apical meristems and intercalary meristems. Next we will move on to what is a periderm. Okay, so this question, what is a periderm? So, in clear note, if I have to tell you, when you are choosing this question, definitely it should be diagram based explanation. In a schematic way, in a systematic way you have to explain. But in brief, if I have to tell you about periderm, okay, just a second now. Yeah, in brief, if I have to tell you about periderm, periderm is nothing but because of the activity of cork cambium which is taking place in cortex. cork cambium, cork cambium, also addressed as phellogen. Fine, right? So this cork cambium will differentiate, okay, the new cells which are formed by cork cambium as a result of its meristematic activity towards the epidermal region are addressed as cork cells or phellum. This is addressed as cork cells or phellum. And those new cells which are formed by cork cambium towards the endodermis, this is towards what? Towards epidermis. This is towards epidermis. And this is towards endodermis. This is addressed as secondary cortex, or phelloderm. Secondary cortex or phelloderm. This is towards what? Endodermis. This is towards endodermis. Here we go. In dicot stems, periderm is the most protective secondary tissue which is formed during secondary growth, which is replacing the ruptured epidermis. Okay, and the components of it, if you see, it consists of phellum, that is cork, phellogen, which is secondary, which is cork cambium, and phelloderm, if you see, that is secondary cortex. Clear, right? So here we go with the tissues in detail. Yeah. So epidermis at the end, followed by complementary cells which are freshly formed cells below the epidermis, which is rupturing the epidermal wall layers, and below that, if you can see, cork cambium and secondary cortex. Above cork cambium you can see these are what? Cork cells or phellum. Okay. This diagrammatic representation followed by sequential explanation is required, how a periderm is formed. Fine, right? Next, if you observe functions, what are the functions of secondary cortex? During secondary growth, epidermis will break and they get replaced by cork cambium which is phellogen. This is formed in which? In the cortex region. Phellogen will cut the cells on the both sides. Outer side, the cells which are formed, these are phellum or cork. This phellum or cork will have suberin depositions above it, which are impervious for gaseous exchange due to the deposition of suberin. And inner side to the cork cambium, if you see, what is formed? Phelloderm is formed, which is parenchymatous. Phelloderm is formed towards the inner side of cork cambium. This is parenchymatous. And together, these three layers will form what? Periderm. Together, these three layers will form periderm. Next, if you observe some regions of phellogen, complementary cells, some regions of phellogen will produce complementary cells which are forming lens shaped openings of called as lenticels. So the openings or the breaks on the epidermis which are formed are lens shaped. So they are addressed as what? Lenticels, which will help in gaseous exchange. There are no breathing organs. We have learnt in the respiration in plants, there are no breathing organs, but then lenticels are present for gaseous exchange. Fine, right? In the stem, like how stomata is there in the leaf, here we have lenticels for stem. Next, you see a transverse section of the trunk of a tree. This shows concentric rings. They will show what? Concentric rings, which are known as what? Annual rings. How are these rings formed? What is the significance of these rings? What is the significance of these rings? This question is appeared in March and in May as well. Okay, questions appeared in March and May. Here we go with this annual rings. Concentric rings which are formed when you see the transverse section of a tree trunk. You can observe the activity of both vascular cambium and cork cambium, which are the secondary activities. Fine, right? Vascular cambium and cork cambium are seen. But most of the secondary growth is conducted by, vascular cambium only. So here if we observe Formation of annual rings. Activity of vascular cambium. What is the activity of vascular cambium? In dicot stems, secondary growth will occur due to the activity of vascular cambium, which will produce secondary xylem, that is wood, and secondary phloem. And if you see the seasonal influence in the tropical plants, if you see, because of the variation of different seasons, the formation of the annual rings also varies. So, the secondary growth is variable based on the seasons. What is the seasonal influence on the secondary growth? You have to mention. Mention your answer, present your answer sequentially. Highlight all the points, all the subheadings and mention. Okay? Then your answer looks impressive. So, here seasonal influence, the activity of cambium is influenced by physiological and environmental factors, especially seasonal variations. Next is spring wood or early wood that is formed. This is during favorable season, that is in spring, where the cambial activity is highly active, and then it produces xylem vessels with wider lumens. Xylem vessels are formed with wider lumens, and thin walls are present which are efficient for water transport. Then autumn wood or late wood, this is formed during unfavorable season in autumn or in winter, where the cambium activity is less active. This produces xylem vessels with narrow lumens. Xylem vessels are formed with narrow lumens, and this thick walls are formed. This is suited for mechanical support only. And then annual combination. The annual band, the band of spring wood and autumn wood together will represent one ring. Remember, the band of spring wood and autumn wood together they form one ring. Concentric arrangement, each year a new annual ring is formed which is outside the previous one. Okay, this will appear what as concentric rings in the transverse section of stem. What is the significance of the secondary growth? By counting the number of annual rings, the age of tree can be estimated. Age of tree can be estimated by counting the number of annual rings. This study of annual rings is addressed as what? Dendrochronology. What is dendrochronology? Where the age of the plant is identified by number of annual rings that are formed. Next, older rings form what? Heartwood, and this is important source for timber. Clear? So, look into the secondary growth. If you are having any doubts, just ask me, ma'am. Next, what is the difference between lenticels and stomata? What is the difference between lenticels and stomata? Lenticels are found in the cork tissue of secondary stems. This is found in cork tissue of secondary stems, and stomata is found in epidermis of leaf, young stems and other aerial parts. Next, you see these are made of, lenticels are made up of loosely arranged complementary cells without any guard cells. And stomata, each stoma is surrounded by two guard cells. Sometimes this is also having subsidiary cells. Next, they will remain open all the time. Lenticels will remain open all the time, whereas stomata will open and close depending on the turgidity of guard cells. Each difference, mention it properly. Okay, both lenticels and stomata, they are used for gaseous exchange, but at different locations, one at the leaf and one in the stem. Lastly, highlight the diagrams as well. Lenticels are common in woody aerial parts of the plant, and stomata is present in terrestrial plants, mostly in the leaves. Here, lenticels will permit gaseous exchange between inner tissues and outer atmosphere. This will help in transpiration, and stomata will regulate transpiration and gaseous exchange in the leaves and in young stems. Here we go with the diagrams in proper. Okay, look at it, and if you are having any doubts, just ask me. Here we go with the next question. Cork cambium forms tissues that form cork. Do you agree with the statement? Explain. Here the same explanation goes for the secondary growth, okay, and the same diagram, but you have to explain it in the sequential way, okay. Next question. Next we will move on to the chapter, that is modes of reproduction. Here we go. What is the clear difference between asexual reproduction and sexual reproduction? We have to distinguish between them. And why vegetative reproduction also considered as type of asexual reproduction? Okay, the questions, if you have seen, this is appeared in 2024, 2020, 2016 and 2015. That important question this is. So, if you have to highlight this, first asexual reproduction and sexual reproduction. Asexual reproduction, there is no fusion of male and female gametes, but here if you see, fusion of male and female gametes occur. Formation of the gametes and fusion of the gametes occur. Here there is no fusion of male and female gametes in asexual reproduction. Single parent is present, so this is uniparental. Single parent will give rise to the offsprings. In asexual reproduction, that is uniparental. If you see, gametes may be formed by some or the other individuals, okay. This two individuals will be involved, two gametes will be involved in the gamete formation, so this will be biparental, okay, biparental. Just a second. Yeah, so sexual reproduction, this is biparental. Fine, right? And then here you see, for asexual reproduction, offsprings are genetically identical, which are known as clones. Here in sexual reproduction, offsprings show genetic variation and these are not identical to parents. These are not identical to parents. Next, here you see asexual reproduction occurs by spores, tubers, bulbs and rhizomes. Sexual reproduction, this occurs by fusion of gametes, and here this is common in unicellular and simple organisms, asexual reproduction. Sexual reproduction is common in multicellular organisms and also in some unicellular organisms. Here asexual reproduction process is simple and rapid. This is comparatively complex and slower process, okay. Highlight the answer with the diagrams. All the time diagram based explanation is important, ma. Fine, right? So, here isogamety. Two gametes are similar in their morphology, structure and physiology. Those two gametes are taken. For example, chlamydomonas. Write a brief account on gametogenesis with examples. Appeared in June 2019 and May and March 2016. June 2019 and March 2016. If you see, how you can give a brief account or note on gametogenesis? This is a process by which diploid or haploid precursor cells will undergo cell division, differentiation to form mature haploid gametes. This will form mature haploid gametes. This is known as gametogenesis. Next, in fungi, algae and bryophytes, the plant body is haploid and the gametes produced by what process? Mitosis. Fine, right? Because the gametophytes are haploid, they will give rise to haploid gametes by undergoing equational division. Next, in pteridophytes, gymnosperms and angiosperms, the main plant body is diploid sporophytic plant body. Sporophytic plant body. Here, special cells will be formed. These special cells are called as what? Meiocytes. These special cells are meiocytes, which will undergo meiosis and they will produce haploid gametes. What are different types of gametes? In some algae, if you see, for example, Cladophora, the gametes are morphologically similar and they are called as what? Isogametes or homogametes. Next, in most sexually reproducing organisms, gametes are morphologically distinct, means they are separate, they are distinct to each other, okay. They are called as what? Heterogametes. Highlight the terms of homogametes and heterogametes, that is important. Next, male and female gametes. The male gamete is called as anthrozoid or sperm and female gamete is called as egg or ovum. For example, you see it in Funaria, Pteris and Cycas. Fine, right? Male gamete and female gamete. These two are homogametes. Best example is Chlamydomonas. These are heterogametes, where the male gamete anthrozoid is biflagellate and the flagella is on the lateral ends or lateral sides, and the female gamete is large and non-motile. Egg or ovum. Examples here you can see isogametes, example Cladophora all algae, heterogametes, Fucus algae, Funaria, Pteris and Cycas. Next, list the changes that are observed in angiosperm flower subsequent to the pollination and fertilization. Here you can see this question has appeared in May 2024 and 2017. What are the changes in angiosperm flower post pollination and post fertilization? So, distinguish them clearly. First, after pollination, what are the changes that you can observe? In over 60% of angiosperms, pollen grains will be shed in two-celled stage only. One will be vegetative cell and the other is generative cell. In some, generative cell will further divide to form what? Two male gametes. Generative cell will further divide to form two male gametes before shedding. So, they will be released in three-celled stage. Next, pollen grain will germinate on stigma forming what? Pollen tube. Pollen grains germinate on stigma forming pollen tube through a germ pore. Next, the pollen tube will grow through the stigma and style tissues. This will reach the ovary. Pollen tube enters the ovule by porogamy, chalcogamy and mesogamy. Inside the embryo sac, the pollen tube will burst in a synergid and this will give out two male gametes. The two male gametes will participate again in double fertilization. Clear till here? Any doubt anywhere? Now we will look into the post fertilization events or post fertilization changes. There are four post fertilization events that happen. One is endosperm development, second is embryo development, third will be seed formation and fourth will be fruit formation. That you have to give a clear picture in post fertilization events. So, first if you see floral parts like calyx, corolla, stamens, stigma wither away or fall. Okay, they will disintegrate, they will wither away or fall. Next you observe ovary will develop into fruit, ovule develops to seed. This happens simultaneously. And then if you see in seed, the outer integument in the ovule will turn to testa and the inner integument of the ovule will turn or form the tegmen. Micropyle, which is at the base, micropyle will transform into seed pore, and hilum, which is the scar of the seed, and funicle will be the stalk of the ovule priorly and funicle will be what? Stalk of the seed later. Clear, right? So, these are the changes, and zygote develops to embryo, primary endospermic nucleus will form what? Endosperm. Synergids and antipodals inside the embryo sac will degenerate. Like that, you have to highlight what are the changes that are happening inside the embryo sac, what are the changes that are happening inside the ovule, what are the changes that are happening into the peripheral non-essential parts as well, you have to mention. Next. Just a second, ma. I will be looking into the life cycles or what are the highlighted questions still we have to attend. Yeah. Angiosperm life. Yep. So, two more questions. Yeah, this one you see. Fertilization is not an obligatory event for fruit production in certain plants. True? Yes or no? Yes, fertilization is not obligatory for fruit formation. Fruit formation without fertilization is addressed as what? Parthenocarpy, which is an important segment for your objective exam also. So, fruits which are developed without fertilization of the ovary, those are parthenocarpic fruits. This phenomena is called as parthenocarpy. This is seen in the plants like banana, grapes and cucurbita. Next, apomixis, you see, apomixis is a type of asexual reproduction that mimics the sexual reproduction because the ploidy at the end of the daughter cells is diploid. So, here some flowering plants, certain species of Asteraceae and grasses, they will produce the seeds without meiosis and fertilization. This special mechanism is addressed as what? Apomixis. This special mechanism is apomixis. Apomictic seed production, you see, in such plants, seeds are formed without fertilization. Seeds are formed without fertilization and fruits may be produced parthenocarpically. Next, asexually they will mature. Apomixis is a type of asexual reproduction that will mimic the sexual reproduction. In some cases, diploid egg cells will develop into embryos directly. This is also without fertilization. So, assured reproduction here you see, apomixis ensures reproduction even in the absence of pollinators. That is one advantage which is advantageous in extreme or unfavorable environments as well. Even when pollinators are not present, the reproduction takes place as usual. Next, identify each part in the flowering plant and write whether it is haploid or diploid. Okay, identify and write whether it is haploid or diploid. Very simple, ma, ploidy type of question. Very simple, you can answer, you see. Firstly, go with a tabulated form of explanation, that goes good. First, you see what are diploid in this. What are diploid? So, first sporophytic generation will start with zygote only, no? The sporophytic generation will start with zygote. Zygote is diploid. Followed by ovary, okay? Ovary is diploid, anther is also diploid. Why? Because sporophytic phase starts with zygote, this is diploid, this is because of the fusion of what? Haploid male and female gametes will give diploid zygote. This forms out the beginning phase of sporophyte. Ovary, inside ovary ovule will be present, inside the ovule only megaspore mother cell will be formed, right. So, all the cells that are present inside the ovary are diploid. This is a part of sporophyte. This is formed from diploid maternal tissue, sorry. Next, anther, if you see, this is diploid. This belongs to sporophyte and this contains diploid microspore mother cells. Now, we will look into the haploid cells. Haploid cells, if you see egg and pollen, these are the haploid. This is haploid female gamete. Pollen is haploid male gametophyte which consists of male gamete. This is haploid male gametophyte. This represents the male gametophyte which are formed by meiosis of microspore mother cells, and male gamete which is from the pollen grains. This is haploid only. This is formed as a result of mitosis of generative cell. Clear till here? So, life cycle of angiosperm is not many times asked, but then I will explain you with the diagram. Okay, phases of the angiosperm, life cycle of the angiosperm. So, first you remember this, ma. The phase, the angiosperm starts with what? Its plant body is diplontic plant body only, that is sporophytic plant body, and the life cycle is diplontic life cycle itself. So, here if you observe first from the zygote formation, if you see, yeah, zygote which is diploid inside the embryo sac and primary endospermic nucleus is triploid. This gets matured to seed and embryo which is formed inside, and this develops into young sporophyte which is diploid. And inside young sporophyte you will see reproductive structures, male and female reproductive structure. Male reproductive structure is what? Which consists of, like it is stamen, it consists of anther and filament. Female reproductive structure is gynoecium which has stigma, style and ovary. Inside ovary, ovule is present. Most of the angiosperms have inverted ovule. Inside ovule, you will have nucellus cells which are diploid. One of the nucellus cells in the micropylar region will get differentiated to megaspore mother cell. Here also, microsporocyte is present inside the anther, inside the sporogenous tissue inside the anther, wherein both the cells will start the division. Okay, first meiotic division. After meiotic division, male gametes and female gametes are formed. This is the haploid phase. Okay, and after the fusion again inside the ovary, pollen pistil interaction, double fertilization, everything we have learnt in sexual reproduction of flowering plants. after which it forms what? Zygote again. If you can see the complete life cycle, all the stages or phases are diploid except haploid pollen grain, which is male gametophyte, and haploid embryo sac, which is the female gametophyte. Except those male gametophyte, female gametophyte, male gamete and female gamete, rest all the stages of life cycle are diploid. So you address the life cycle of angiosperm as diplontic life cycle. This is the complete account on angiospermic life cycle. In our next class, we will look into the sexual reproduction of flowering plants question. So that's all for today's session. If you are having any doubts, please drop your messages in the comment section. I'll address all your doubts. Okay. So we'll meet in the next class with new set of questionnaire from sexual reproduction of flowering plants. Thank you.